My name is Maria Cristina Schilling Costello. I am working on Occurrence and Fate of Contaminants of Emerging Concern.
So my research focused on contaminants in water from two major rivers in Pakistan. Now, instead of shipping water samples to the US, what we were able to do was to take samples and pass them through a matrix. In this case, it was an HLB matrix in a solid phase extraction cartridge. Now what this matrix does is when you pass water through it, basically it will grab onto the compounds. So with all the compounds in this cartridge, they were then able to ship the cartridge to me. So I was able to extract these compounds from these cartridges under vacuum in this hood. So what happened then is I ended up with these compounds in an organic solvent. In order to analyze even smaller concentrations, what we do is we actually flow down the samples and evaporate the solvent with nitrogen gas.
Samples were then injected into a liquid chromatography system with a specialized mass spectrometer that allows for compound identification and quantification. We targeted quantification of 68 organic compounds. In general, seasonal differences were typically observed, with compound concentrations being low under high flow conditions. High flow conditions resulting from snowmelt tend to dilute chemical concentrations, since these sources are unaffected by anthropogenic activity. Heavy rains during the monsoon seasons could bring in additional chemicals such as herbicides and other chemicals used in agriculture from the soil being eroded into the river. But this may not lead to an observable increase in water concentrations due to the dilution effects from the massive amounts of water. The compounds that we found and the concentrations we found varied at different river locations. A group of endocrine disrupting compounds known as plasticizers called phthalates were found at all locations along both river systems. Bisphenol A, another plasticizer known to disrupt the endocrine system, was detected in most samples, but was at lower concentrations than the phthalates. Several pharmaceuticals, including sulfamethoxazole, diclofenac, ibuprofen, acetaminophen, and naproxen, were detected at some locations, but consistently at lower concentrations. In the Swat River, total phthalate concentrations ranged from 100 to 500 parts per trillion during high flow and 500 to 4,300 parts per trillion during low flow. Total pharmaceutical concentrations range from non-detect to 20 parts per trillion during high flow and non-detect to 820 parts per trillion during low flow. Using heat maps, a data visualization tool, where the yellows and reds refer to higher concentrations, whereas the blues refer to lower concentrations, the differences between upstream and downstream measurements can be seen, as exemplified here for the data in the Swat River during the low flow river season, where concentrations downstream are higher, as noted by the red and yellow colors, compared relative to upstream, as noted by the blue color. In the Kabul River, total phthalate concentrations range from 200 to 900 parts per trillion during the high flow season, and 400 to 1,000 parts per trillion during the low flow season. The total pharmaceutical concentrations range from non-detect to 10 parts per trillion during the high flow, and 800 parts per trillion during low flow. In the Kabul River, seasonal differences in phthalate concentrations were only observed at a few sampling points. Whereas for pharmaceuticals, seasonal differences were consistently observed, with pharmaceuticals being primarily detected only under low flow conditions. The map showing low flow data for phthalates and pharmaceuticals in the Kabul River did not reveal any clear trend in differences between upstream and downstream, unlike what we observed in the Swat River. The phthalates that were detected in all the river systems were often at higher concentrations than the other organic compounds we targeted for quantitation. Phthalates are known to mimic hormones, thus disrupt the endocrine system that controls sex differentiation in fish, as well as several other health indices. However, based on data from the United States Environmental Protection Agency knowledge base, the phthalates and bisphenol A concentrations in both the Kabul and Swat rivers are below levels expected to produce an observable effect in fish.